Hi, Julie. Hi, Sarah. I figured I'd pop into the DNA subway blue line to kind of give you a little tutorial about what's going on here. So this is the alignment viewer, right? And here's our consensus. So the consensus tells us this is, based on what your species is, this is what you is normally seen, right? And these colors represent the nucleotides that are here. So green is an A, an adenine, T is thymine, C is cytosine, and G is the black is a guanine. Okay, so what we do is we compare everything back to the consensus. Okay, so if we look at this sample right here, I believe this was site 3, right? Cali site, this is O38. And so we can see that there are some differences here. All right, now the gray means things aren't lining up at all. So we can go back and deselect 18. I believe O18 is not a mosquito at all. It's something else, and that's why it's here, right? So we can actually get rid of that and kind of trim this out to make it a little more understandable. But basically, when we look at the consensus here, whatever is expected is in the consensus, and then this is slightly different as far as the base is concerned, which you can actually see if you switch this out, right? And so this shows us what some of the bases are here. These are unreadable, right? But we can see the differences. So in the consensus, this is a T. However, for numbers 17, 2, and 35, it's an A, right? And this is supposed to be a T. There are C's that are here. So we can see some variation in the gene sequences, right? And so we call these variants. It doesn't necessarily mean that there's a mutation. It doesn't mean that it's a different species other than what it's identified with. But that's some of the information that we use in your findings to go back and say, okay, here's what's going on in this environment, and we can see some of the variants here. Okay? So that's what that means. So we can kind of come back here, and if you've pulled up the sample papers that I've sent you, you'll see how they use the alignment viewer, right? That's here, to actually make a case for your findings and looking at some of those differences, okay? And always keep in mind that when you look at your findings, you want to compare it back to your original hypothesis based on the locations of where you collected your samples from, okay? So I'm just going to X out of this for a second because we can go back to select data anytime that you want. So I'm going to take out 18 and I'm going to save that selection, and now I'm going to remuscle it. Okay, so after you remuscle, you can open it back up. You see the alignment viewer, you see 18 is not here, it looks a little bit cleaner. So you can decide based on what these species are. I would keep a running list on a table on a piece of paper that identifies the species name to help you interpret the data. So O38 is what species, O17 is what, and then also consider the locations that you caught them from, right? So that's some of the things that you want to present in your findings. All right, another useful tool is to come over here and use the sequence similarity, right? So it kind of sets up a correlation table. And this is something that Anson and Justin actually used very effectively in their papers. So if you don't have that particular paper, I can show you how they wrote those results. But like we can compare 1 to the consensus, right? We can compare 2, and that means the 2 over here, O17, to the consensus. And you can see the sequence similarity in that, right? So these are over 90% here, right? So we're pretty certain for what we're seeing. However, if you compare 2 to 1, right, only 79%. Okay, so these sequence similarities are also useful. You can take screenshots of these as well and include them as figures in your papers. All right, so I hope that gives you a little more of a clue about what to do with these alignment viewers. These images should definitely be placed into your results. And you can notice, I think Emma did this as well, like you can choose the data. So what you can do to make it more understandable is like if you know the locations of your stuff. So for example, we've got 2, 5, I'm going to take out 17, right? I'm going to do this based on what locations they were in. So like I happen to know that 2 and 5 were from location 1. So I'm only going to select those two. And note, I'm not doing any of the blast hits down here. Okay, so I'm going to save those selections. I'm going to come and muscle that. And then when you open it, you're only looking at those, right? So now we know this is location 1. You can put the species name that was identified from the blast hits here. And then you can do you can take a screenshot and you can do the comparisons, okay? You can also do that with the sequence similarities here too in a very small table. All right, so I would do that. I think it would make it more understandable for you to interpret the results as well as your reader in the paper in your results section. Okay, so and then anytime you want to go back and switch that up, you can do that. So like we know that this is location 2, I believe, up to here. Pretty sure, right? Jules, I think this was yours. We'll save those, muscle it, and here it is, right? And just remember you're comparing everything back to that consensus. Of course, I left 18 in there. It doesn't match anything, and we know that because it was not a mosquito. Okay? So that's how you would do it. You can look at the sequence similarities here as well and include those as separate figures. I think that would be a great thing to do. I do believe Emma set her paper up that way, and perhaps Ethan as well. I'm not exactly sure. So take a look at those. Let me know if you have any questions. Once you've muscled everything, you can actually, based on the data that's selected here, you can create the phylogenetic trees. And if you look at the PDF that they sent you, okay, so here's the muscle alignment. I just showed you that. The generate the tree, so this is on what, page 21? So you can create the phylogenetic trees over here. The neighbor joining tree is there, so that you can see how the species would be related to each other on this particular cladogram. Okay, so let's try that based on what we just did. We'll open it up, right? And so we can see how closely these are related. So for example, they're showing the differences in the sequences here, right? And this suggests a most recent common ancestor back this way, right? So it's telling us that 29 is more closely related to 17 and 26 is more closely related to 18, and that 27 out here is an outlier, right? It's completely different species. So that's something you can also do. We can do it in max likelihood as well. And so this just puts some probability in there. And this supports our findings, right? So like here's 18, total outlier, where this is concerned, okay? And so here's 17, here's 29, a little more closely related, putting 27 back based on the DNA sequence analysis of the recent common ancestor. Okay, so again, you can switch those up at any time by selecting the data. So if you want to use all of it, why don't we just do that? We'll put all these samples in, we'll save our selections, we'll remuscle it. Now we can come back down here to this first one, right? Again, that's the neighbor joining tree. The tree opens in a new window, of course, and so we can open that and see it. And then this shows you some of the relationships between your species, right? So the tree is telling us 37 and 29 closely related, and again, you can you can print these out. Take a screenshot, print it out, and then to help you interpret it, put the species name here. And then you can go back, and if you go into your sample database and you log in with your information, this is where you can look at the pictures of each of the samples that you collected and compare it. You can compare the morphologies or the anatomies of them. Where are they similar and where are they different, right? Okay, so this one's showing 2 as an outlier all the way back here. The most differences. Okay. All right. So I hope that's helpful to you. Let me know if you have any questions. I'm happy to help you at any point.